So today I'm going to talk about target therapy of neuroblastoma with a cell-penetrant ATF antagonist peptide. And I do want to thank Hyundai because they've actually supported my research for the past several years. And actually, the first grant that Hyundai supported was looking at a synthetic lethal screen in MYC amplified neuroblastoma. And as Kim pointed out, MYC amplification is a very poor prognostic marker for neuroblastoma, but it's very hard to target. So we went about trying to actually look at MYC neuroblastoma by doing a synthetic lethal screen. So we took a cell line called SHEP21N, which had MYC under a control of a TET-regulated promoter, and then we infected that with a shRNA library, which targeted over 18,000 genes. And so you can look as far as, we were looking for genes that when MYC was on, that was synthetically lethal. And from this screen, we actually identified about 218 genes that were significantly depleted. So the first gene that we actually identified, called TFB4, actually my graduate student Shobo Zhang were wrapping up that project. But the second gene is a gene called ATF5. So ATF5, or otherwise known as ATFX, is a member of the ATF cyclic AMP responsive element binding or Krebs family of transcriptional factors. And it's involved in a wide variety of cellular processes, including cell cycle progression, metabolite homeostasis, cellular differentiation, and apoptosis. Fortunately for me, actually, Lloyd Green, who is actually one of my mentors at Columbia, and Jim Angelastro, who is currently now faculty at UCSF, have actually been studying ATF5 for the past decade or so. So in 2003, they described that ATF5 is expressed in neural stem cells and progenitor cells, but not really in mature neurons. So this is a photomicrograph showing nestin, which marks for neuronal progenitor cells, and you can see sort of the nuclear ATF5 staining. In contrast, mature neurons, and you can see sort of the nice neurites that are here, you actually don't see ATF5. So they next went on to say, okay, what happens in sort of the very primitive cells? Can we look actually at GBM? And that's actually what they looked at next. And in 29 GBMs, they actually saw expression of ATF5 in every single one of the GBMs, and actually the vast majority of cells actually had nuclear expression of ATF5. So this is a study from Michael Green at University of Massachusetts, in which they found that ATF5 actually correlates with poor prognosis in malignant glioma. And ATF5 has also been described to be actually expressed highly in other cancers, including breast cancer and pancreatic cancer. So what about neuroblastoma? So we initially looked actually from the oncogenomic side to see if ATF5 expression correlated with poor prognosis, and we find that there was a very nice correlation with high expression with overall poor prognosis. We then looked at data from Kosak et al. Looking at ATF5, and this is the stage 4 MYC amplified cases, where in the red, where there's very high expression, and significantly different from the stage 4 non-amplified cases versus the stage 1. We have similar results looking at the target cohort as well. So it really appears that ATF5 correlates with MYC amplified neuroblastoma. And this is just some very preliminary data looking at immunostaining for ATF5, and you can see sort of the nice nuclear staining in this patient with stage 4 neuroblastoma. So functionally, one question is what does ATF5 do? So if you actually knock down ATF5 in 2 different neuroblastoma cell lines and do what's called a colony formation assay, and you knock down ATF5, you get a marked inhibition as far as colony formation for one cell line called SKDNZ, and also a cell line called BE2C. Knocking down genes is actually pretty difficult, so the question is can we do this in a different way? And so my colleagues, Jim Alejastro and Lloyd Green, have actually developed a dominant negative ATF5. So they took ATF5, they internally truncated it, and in that instance, it lost the DNA activation and binding domain, but still retained actually the B-zip leucine domain. So it can actually bind to the endogenous ATF5, also the heterologous partners, but it can act as a dominant negative inhibitor. And so they used a retroviral delivery system, and this is actually a C6 glioma model in which they actually injected C6 glioma cells into the rat brain, and then they retrovirally injected this dominant negative ATF5, and there was a marked increase as far as apoptosis. In fact, there was almost 100% apoptosis within these C6 glioma tumors. If you look then at neuroblastoma, we can look at the same thing, and this is doing it in vitro. You can actually get some nice apoptosis occurring. But as was sort of discussed before, retroviruses have their problems. They can induce secondary malignancies, and it really isn't a practical way to actually try and deliver a peptide or a drug. Back in about 20 years ago, the Drosophila antennapedia homodomain was noted to actually be internalized by neuronal cells. And from that, they actually identified a short peptide called penetratin, which actually allows this peptide to actually penetrate into the cells. Now, it may do it in a couple of different ways. It may be endocytosed, but actually then can get into the cytoplasm, or there may be direct sort of pore injection. Another sort of HIV TAT protein peptide can actually also penetrate into cells as well. So what we can do is we can attach the penetratin to this dominant negative ATF5, and we can either produce it in bacteria and purify it, or fortunately we can also synthesize it as well, although I have to say it's a little bit more expensive to do it that way. If you look in vitro at least, you can look at neuroblastoma cell lines, and if you take this dominant negative ATF5 with a cell penetrating peptide, you can actually markedly inhibit actually the neuroblastoma cell lines. And so for the most exciting data that we actually have, or I should say that we can see, is this actually also works in vivo. And so if you have a glioma cell lines, and this is sort of a mouse brain in which there's a tumor here, and you actually give this subcutaneously, not actually directly injection, which was how the retrovirus was done, we can actually have marked apoptosis occurring with, which is here, within this tumor. And the flag tag is just where the vector is. And there's marked inhibition as far as tumor growth. So to conclude, I'll talk about what our specific aims are. We're going to look how this cell-penetrating dominant negative ATF5, how it selectively kills neuroblastoma cells. We're going to look as far as the apoptotic mechanisms, identify synergistic pathways, and can, in fact, does it induce neuronal differentiation. Lastly, we're going to do this in vivo. We're going to actually try and target both the xenograft tumors, and we're also going to try and see whether we can inhibit the Bill Weiss's TH-MYC model of neuroblastoma as well. So with that, I'll conclude. I'm trying to zip through this, if you would, is to say thank my people in my lab, Shobha Zhang and Debarshi Banerjee, my collaborators Lloyd Green, Marcus Sigilin, and Jim Lastro. And lastly, I also thank Hyundai Hope on Wheels. So thank you very much.